All right, in today's lecture, we're going to talk about the process of photosynthesis. Remember, it's essentially an opposite process to cellular respiration, which we've talked about previously. And the goal of it is to take carbon dioxide and water and using the sun's energy, create sugar, create food for the plant. Now remember, plants do respiration. Okay, some people think that plants do not do respiration. In fact, they do both respiration and photosynthesis. It's just the way plants get their food is they know how to make it versus, and they're called autotrophs versus heterotrophs, which have to go out and get their food themselves. Okay, so quickly just take a look at the relationship between photosynthesis and respiration. I just mentioned they're opposite processes where the inputs of one are essentially the outputs of the other. In the case of cellular respiration, you take sugar, you break it down using oxygen into smaller and smaller parts, eventually releasing that as carbon dioxide. The electrons end up transferred on water, and we create molecules of ATP through that process, which we can use for cellular energy. Plants will do the opposite of that. They'll take water and carbon dioxide and using energy from light, take those and fuse them together. And we're going to talk a little bit about that process, inevitably making glucose. So the term for organisms that do photosynthesis is photoautotrophs. Photo meaning light, autotroph meaning makes their own food. Another term that's commonly used, we'll talk a little bit more about this term in ecology, is producers. So they use light to make food. Examples of these things are your common plants, algae, some single-celled protists, which are single-celled eukaryotes, and there are also some bacteria that are able to do photosynthesis. Remember, that's in contrast, as I just mentioned, to heterotrophs, which cannot make their own food. They have to go out and get it from their environment, and they are called, instead of producers, they are called consumers. We'll talk a little bit about this. So there are these things called chemiautotrophs, which make their own food, but instead of using light, they'll use chemicals and the reducing power of certain chemicals to do so. So where does this process occur in the plant? What organelle? Specifically in the chloroplasts. Now chloroplasts are similar but not exactly the same as a mitochondria. Obviously they're green. They consist of 3 membranes instead of just 2 sets like the mitochondria does. There's an actual interesting explanation of why that is, but we'll get to that later. So it has an outer membrane, an inner membrane, and then on the inside it has thylakoid membranes. And it's those thylakoid membranes where a lot of the magic happens for photosynthesis, specifically the first reactions of that process. The part that the thylakoid stacks sit in, or the grana sit in, as we'll talk about in a second, is called the stroma. That's the liquid like cytoplasm within it. And then the thylakoids are the things that look like pancakes, those stacks of coins that are on there. You do not have to be able to differentiate between thylakoids and grana, but just know that both of them can be used to describe the same thing, the membrane where the kind of the important reactions happen in photosynthesis, or at least some of the important reactions. So just so you can have a quick look here, we have going all the way down to the organelles, right? This is a chloroplast, right? This is the cartoon version of it. And if we look into there, these are showing individual chloroplasts within a cell that does photosynthesis. And we zoom in, or zoom out a little bit further, we can see a cross-section of the leaf where these cells exist. And then we zoom out even further, you end up having the leaf, which is one of the organs. Probably never thought about that, but it's one of the organs of a plant, or a tree in this case. So photosynthesis summarizes this process. We take carbon dioxide, which we're going to go into this, which is called the Calvin cycle, the dark reactions. And we take water, which is split, which is actually our source of electrons. And using light energy, we are going to generate glucose, oxygen gas, and some water too, but not as much as we put in. Okay. Now, photosynthesis, just like respiration, is a redox process, an oxidation-reduction reaction, but it's the opposite. In this case, water is oxidized. Remember, in respiration, water was the thing that took the electrons at the end, so it was reduced at the end. Oxygen gas gained electrons and hydrogens and formed water. In this case, we're going to do the opposite process. We're going to take those electrons off of the water, so we oxidize the water, And we're going to eventually put them on carbon dioxide, and when we add electrons and free energy to the carbon dioxide molecules, and we put a bunch of carbon dioxide molecules together, which is our source of carbon, we are going to make glucose from that. So because carbon dioxide gains those electrons, it is reduced. The two goals of photosynthesis are to use light to create high energy molecules, and then to use those high energy molecules to not to fix carbon, but to take fixed carbon and create sugar with those high energy molecules. They provide the free energy that we want to store in the sugar itself. And that happens through two main reactions: the light-dependent reactions, sometimes just deemed the light reactions, and the light-independent reactions, sometimes deemed the dark reactions. The light reactions are going to occur in those thylakoid membranes, which is, I said, why they're so important. And then the dark reactions are going to occur in the stroma. Now, before we can talk about how photosynthesis actually works, we have to understand the nature of sunlight itself. Now remember, sunlight is a type of electromagnetic energy. It travels in waves via a process called radiation. There are different types of electromagnetic radiation, including infrared, UV, X-rays, gamma rays, microwaves, and radio waves. And visible light is only a very small part of that overall electromagnetic spectrum. Plants tend to use visible light in order to make food. Now, these other types of radiation are important sometimes. Like, for example, UV radiation can actually cause DNA damage. X-rays, anything usually below or with a shorter wavelength than the visible light spectrum, can cause DNA damage. Anything above that does not, like infrared's another word for heat radiation. Microwaves, again, that doesn't cause mutations, it causes water molecules to vibrate. That's why microwaves work. Radio waves, which are passing through our bodies all the time. So visible light is just a small part of that spectrum, and our eyes also use visible light in order to kind of differentiate between things in nature. So a pigment is anything that absorbs visible light. Any light that it does not absorb, it will reflect. So for example, my shirt is blue. The reason it appears blue is because it's absorbing other pigments and reflecting blue back at you. So it does not absorb blue. When we're talking about photosynthesis, we want to absorb the color that we are going to, colors that we're going to use for photosynthesis. So let's think about plants. Plants are green. What color do plants absorb? They do not absorb green, they reflect green back at you. So they use reds and purples. If you were to put green light on a plant, it would die. It does not use that type of energy in order to make food. Okay, so remember that idea. It's very important to understand. The reflected light is not absorbed, right? What you see when you look at a pigment is what it does not use. So here's showing you what's called an absorption spectra. The AP Biology exam loves absorption spectra. And so basically what this shows, high peaks are where it absorbs light, and low valleys is where it reflects. Now notice what's reflecting in this. This is actually the different pigments that are found in green plants. You can see that chlorophyll A, chlorophyll B, and carotenoids are all in kind of the absorbing in the purple and blue area, and also in the red and orange. But what's reflected are those low areas, right? The greens and yellows, which is what plants appear to be. They ask you to interpret these graphs. So for example, where there's high peaks, right? That's where there's going to be the most photosynthesis going on with those light, those wavelengths of light. So there's much, it's actually, red will provide it, but it's actually better to do photosynthesis in the purple and blue wavelength area of visible light. This is what chlorophyll A actually looks like. This is the main photosynthetic pigment, and chlorophyll B is an accessory pigment. In terms of their overall structure, they're not very different. I want you to try to figure out what macromolecule type this is overall. Notice the long hydrocarbon chain, or tail. This is a lipid, and this embeds itself into the thylakoid membrane itself, which is why, via its hydrocarbon tail, it's very similar to a phospholipid in its structure. Okay? The difference is this happens to absorb and reflect light. So here below is an absorption spectrum for an unknown pigment molecule. What color would this pigment appear to you? I'll give you a second to answer it. All right. Remember, you wanted to look down at the lowest peaks. In this case, it would appear more blue to you because it absorbs in the violets and the reds, and its lowest valley is in the blue, which is where it reflects the most light. Now, I put this up here for a reason. Then try to think about, like, what am I showing you here? Have you ever had this in your room, for example, on the ceiling? These are glow-in-the-dark stickers, essentially. Sometimes they're made of plastic. You put them up on your wall. But I'm showing you this for a reason. How does glow-in-the-dark work? You may have learned this in chemistry. Essentially what happens is when light is absorbed by the pigments in glow-in-the-dark things, electrons jump up to a higher energy level. And as they fall back down to the ground state, they release photons, energy in the form of photons. Sometimes heat, sometimes photons. In this case, photons. So what they'll do is as they're slowly going back down to normal, it's ground energy state, photons of light are released. That's why you can charge them up by adding light, excites the electrons, and then eventually it will run out of energy and they'll go back to being dim again. So why did I tell you that? Chlorophyll does the exact same thing. When chlorophyll absorbs light, it takes electrons and excites them to a higher energy state. And if we were to just take chlorophyll by itself and not include the other parts of the reactions we're going to talk about, as electrons fall back down to the ground state, it would release fluorescence, which is light, and heat. Okay? So from an excited state, it's unstable, it's going to fall back down to its ground state and release energy in the process. But instead, in photosynthesis, we're going to take that energy and we're going to harness it through several chemical reactions. Okay, so the light reactions, which are going to use this process, occur in the granostacks, or the thylakoid membranes. Okay? The goal is to take water, rip the electrons off of it, those same similar electrons that were put on it in the first place in cellular respiration. We're going to release oxygen, which is exactly the opposite of what happened in respiration. And we're going to produce two high-energy molecules, ATP, which we've heard of, and NADPH, which we've kind of heard of before. That is a electron carrier. But notice, there's no sugar yet. We have not made sugar in this first step. We're going to do that in the next step. Okay? The products of those reactions, the ATP and the NADPH, the high-energy molecules, are going to be used to build sugar later. So here's showing you the basics behind the reactions themselves. Now there are two photosystems to this reaction, photosystem 1 and photosystem 2. You can see photosystem 1 is right here and photosystem 2 is right here. What's going to happen is essentially electrons are going to get excited. I'm going to repeat myself several times because this is a little complicated, but electrons are going to get excited by light. They're going to go to a higher energy state and they're going to pass from complex to complex, similar to how it happened in respiration. In doing so, as we pass from complex to complex, we're going to pump hydrogens across the membrane, just like in cellular respiration. Those hydrogens are going to flow back across ATP synthase, which is the same ATP synthase in respiration, but just this one's found in the chloroplast, and it is going to generate ATP. The difference is this ATP is not going to be used to do, for cellular processes, it's going to be used to build sugar. What happens in photosystem 1 then is the light excites the electrons again. So they got excited in the second photosystem, and now they excited again in the first one. The goal this time, though, is to pass those high-energy electrons to an electron carrier called NADPH. The way you remember that, pH, photosynthesis. NADH is in respiration. pH in photosynthesis. So how does this work? So a photosystem actually is a bunch of side pigment molecules, eventually leading to a reaction center, and all energy that bounces off these pigment molecules eventually makes its way through resonance, which is way more complicated than we can get into. It has to do with quantum mechanics. Eventually ends up on this reaction center where there's electrons waiting. Those electrons are going to jump up as soon as that energy hits them to a higher energy state. Now, this is kind of showing the same thing we just looked at in a slightly more simplified form. The light comes in and bounces from complex to complex, eventually exciting electrons to a higher energy state. Those electrons are passed from complex to complex. Remember, this is located in the membrane. I'm going to show you a little bit better picture of this in a second. Pumping hydrogens across, and the goal is to create ATP through the process of chemiosmosis, similar to in cellular respiration. Eventually, the electrons are going to end up on P700, or the first photosystem. Light's going to come in again, excite those electrons, pass them on to NADPH, creating an electron carrier. So from this process, we create ATP and NADPH, both high-energy molecules. The electrons that we got for this process were taken through the process of splitting water. Remember, so water is the initial source of the electrons. Remember, we talked about where the electrons ended up in respiration. They ended up on oxygen, forming water. Now we're taking water, ripping electrons off, and forming oxygen. Kind of the exact same process, except the opposite. And it's done using light. Here's showing you what it looks like in the membrane itself. Here's showing you photosystem 2 and photosystem 1, the cytochromes. But notice hydrogens, instead of getting pumped in respiration, the hydrogens were pumped up out of the matrix and into the inner membrane space. This time the hydrogens are pumped in to the thylakoid, and they're going to end up flowing out of ATP synthase back into the stroma where the ATP is generated. Again, if they're excited again, end up on NADPH. So again, two high energy molecules, ATP and NADPH. Notice water is split into oxygen, and that's the source of these electrons which get excited by the light. Just as I said before, ATP is generated through oxidative phosphorylation, the term of going and taking hydrogens and pumping them across the membrane, passing electrons from high energy so they end up with lower energy, and eventually having them flow back across ATP synthase and generating ATP. Here showing you one more time, just the same thing. Okay, this is a thylakoid, which are like those stacks there. Here's showing you the light coming in, exciting electrons, which we got from water, forming oxygen. After it's split, electrons are passed, protons are pumped. What do you think is true about the pH in here? What happens to the pH is this pH is going to lower over time because we're pumping hydrogens in, we're increasing hydrogen ion concentration, therefore we're lowering the pH. And as they flow back across ATP synthase, the pH in here ends up rising, actually. It becomes more basic because you're pumping hydrogens out. So when the lights shut off, right, when there's no light, when the sun is down, this actually becomes more basic than it is in the light because there's no hydrogens being pumped into it. So this, you may have noticed this. Why is photosystem 2 first? It was actually discovered second. That's why they call it photosystem 2, even though it happens first. Photosystem 1 was actually discovered first. Photosystem 2 is sometimes called PS680. That's the wavelength of light it uses in order to excite electrons, and PS1 is PS700 because that uses that particular wavelength of light to excite electrons. Remember, water is the source of those electrons, and oxygen is released when water is split. Sometimes they'll talk about radioactively labeling, like water, and where's the oxygen in water, and where's that going to end up. That radioactively labeled oxygen from water is going to end up in oxygen gas. We're going to talk about, well, what if we radioactively label carbon dioxide, the oxygen and carbon dioxide? Well, that's going to end up in sugar. We're going to talk about that later. So the excited electrons move from PS2 to PS1. Again, they get excited again in the first photosystem, and they end up on NADPH. Right? So I'm going to repeat this one more time. Students always struggle with photosynthesis. The goal is to excite electrons and create high-energy molecules. We excite those electrons using light. Light passes from complex to complex, exciting electrons that came from water. Water is split, forming oxygen. Those electrons get excited, and they're passed from complex to complex, eventually pumping hydrogens across into the thylakoid. Those hydrogens flow back across ATP synthase and generate ATP. The electrons get excited again in photosystem 2. But this time their fate is to end up on NADPH, another high-energy molecule, but it's an electron carrier. Sometimes plants, if they have enough sugar, they can do something called cyclic electron flow, where they kind of avoid photosystem 2, and they just keep exciting electrons in photosystem 1, but instead of putting them on NADPH, they put them back through the electron transport chain, pumping hydrogens across, generating ATP after ATP after ATP. Now, if you compare and contrast chemiosmosis in chloroplast and mitochondria, now this is a great essay question, right? Or extended response, I should call it, because there's not really essays in AP Bio. But like comparing and contrasting chemiosmosis in both processes, because they both do it. It just does it in slightly different ways. Like each generate ATP through chemiosmosis. They sometimes use different forms of energy or sources of energy to accomplish this, though. Chloroplasts will use light to excite electrons, right? Mitochondria use electron carriers generated from breakdown of glucose. Those electrons are already high energy when they get them. They don't have to be excited. And we use the energy of those to eventually pass free energy onto ATP in cellular respiration. Now, if you look at the concentration of hydrogen ions in both, notice in the inner membrane space of the mitochondria, we have a higher hydrogen ion concentration, therefore a lower pH compared to the matrix of the mitochondria. In the chloroplast, the higher hydrogen ion concentration is actually inside the thylakoid when the light reactions are occurring. Therefore, when electrons, or when light is there, we have a low pH inside of the thylakoid compared to the stroma. They're both redox reactions. They both involve using and passing electrons to things, right? Both generate hydrogen ion gradients. Both use ATP synthase and generate ATP through proton-motive force, which is the spinning of the ATP synthase protein. So now, remember, the goal of this whole thing, though, is to produce sugar, right? That's what the goal of this. Photosynthesis is to make sugar. And that happens in the second step, the light-independent reactions, or sometimes called the dark reactions. They used to call them the dark more often, but we don't call them that anymore because it kind of creates a misconception that these only happen in the dark. These happen all the time, but they do not have to happen in the light. But they do require the things that were created in the light reactions, the ATP and the NADPH, in order to have these reactions happen. We are going to use the energy from the ATP and the electrons from NADPH to reduce carbon dioxide and form a high-energy sugar molecule. Another term for the dark reactions is the Calvin cycle. Don't confuse that with the Krebs. Everyone always does. It's a different cycle, and it's basically an opposite process. It's going to take carbon dioxide, and it's going to fix it and turn it into sugar molecules, as opposed to the Krebs cycle, which would take sugar molecules, or acetyl-CoA or pyruvate and acetyl-CoA, and break it down into carbon dioxide and extract the energy from it in the form of electron carriers. So the Calvin cycle occurs in the stroma. That's, remember, the liquid part, like the cytoplasm of the chloroplast, and it will form sugar from carbon dioxide using the energy from ATP and the reducing power from NADPH. There's three main phases to the Calvin cycle. There's carbon fixation, there's reduction, and then there's the regeneration of the CO2 acceptor. Now, you don't have to know about that third step as much. You used to have to understand a little bit more. What I care the most about are the first two. The carbon fixation step, which means you take carbon dioxide out of the air and turn it into an organic form. That's what carbon fixation means. And then reduction, which is the process of adding electrons and hydrogens to that carbon dioxide to make it a sugar. The process of carbon fixation occurs in an enzyme, the most abundant enzyme in the world, which is called rubisco. There's tons of it found in every single plant cell. And it's the most, as I said, by far, like it's almost inconceivable of how much of this enzyme exists on this planet. And it's called rubisco. It's basically an acronym for ribulose bisphosphate carboxylase dehydrogenase. Ribulose bisphosphate is the one of the substrates. And it's going to take carbon dioxide or oxygen, and we'll talk about why that becomes an issue later, but carbon dioxide, and takes it and puts it on this molecule, ribulose bisphosphate. You don't have to know that, but you do have to know the name of this enzyme. Okay? It's going to take CO2 out of the air and fix it to an organic molecule, making it organic. And this is the process. Now notice, this isn't too it looks somewhat complicated, and the real thing is actually more complicated than this. This just gives you like little circles to represent carbons. But notice you take 3 carbon dioxides and put them in, and you spit out a 3-carbon sugar. Perfect. So 3 carbons go in, a 3-carbon sugar comes out. In doing so, we use ATP and NADPH to add free energy to and reduce those molecules. Now notice the output of this is not glucose, it's G3P, which is a 3-carbon sugar, similar to pyruvate, but not exactly the same. Remember, pyruvate was a 3-carbon sugar. We then just take 2 G3Ps through a separate process, and we generate glucose from that. So when I said earlier, like if you radioactively label the oxygen on carbon dioxide, it's going to end up into G3P and eventually into glucose, and other organic molecules, because G3P can be used to make amino acids, it can be used to make fatty acids, things like that. So how does a plant get rid of oxygen? Sometimes, because it uses some of that oxygen for cellular respiration, because it does cellular respiration, and water. And how does it take into carbon dioxide? It's kind of like the opposite of us, right? We breathe in oxygen, we breathe out carbon dioxide. They breathe in carbon dioxide and breathe out oxygen, kind of. So they use these little pores in the bottom of their leaves called stomata. And if you had Mrs. Beback, I know she does a lab where you kind of look at this, and I know she focuses on these. They're little pores at the bottom of the leaf, like little leaves, like little mouths. And those pores are surrounded by things called guard cells, and guard cells will open and close depending on the conditions outside. Is it too dry or is it too hot? If it's too hot, they'll close to prevent water loss. And there's actually a really interesting homeostatic mechanism that involves pumping potassium ions into it in order to cause it to open. So during the day, potassium ions are actively pumped into the guard cells, lowering the watering potential, causing the influx of water. When that happens, it's going to cause them to open up, and the stomates will open because of that process as water flows in. So like more water in there equals it opening up. When there's less water, it closes. So when it gets hot and dry, they'll pump the potassium out, and the sodium will, or excuse me, the stomates will close. Remember, this has to do with water potential and things like that. So you could see them kind of tying in maybe photosynthesis with water potential in the AP exam. Who knows? So when the plant starts losing water, the stomates are going to close. But there's a problem with this. When the stomates close, it's like when you close your mouth. And if you plug your nose in your mouth, eventually you start to panic on the inside almost, because you're running out of oxygen and you're building up carbon dioxide levels. The same thing happens with plants, that they're going to start to build up oxygen and they're going to not have enough CO2 to fix into sugar. And the result of this is going to be a phenomenon called photorespiration, which is bad. It's not a good thing. So what happens essentially is, and I told you that the name of rubisco is ribulose bisphosphate carboxylase oxygenase. It's called oxygenase 2 because it also binds oxygen to it, not just CO2. Oxygen is actually a competitive inhibitor of CO2. And if there's a lot of oxygen present and not a lot of CO2, it's going to be more likely to fix oxygen instead of carbon dioxide. And the problem with that is it is a complete waste of energy. When oxygen binds, we don't make sugar, we make molecules that need to be actually broken down. They're useless. And that actually uses ATP. So it's a waste of energy, and a plant will die if it continues to do photorespiration. And this shows you the difference. When CO2 is fixed, we create sugars. When oxygen is fixed, we have to get rid of ammonia, we have to get rid of carbon dioxide, and ATP is actually used, the waste of energy. So obviously on hot, dry days, this becomes an issue because you run out of carbon dioxide and you increase your oxygen concentration when the stomates are closed to conserve water. So what do plants do to combat this? They have certain adaptations. So plants in really hot, dry environments will have adaptations in order to stop this from happening. They'll minimize the effects of photorespiration. There's two you have to understand: C4 photosynthesis and CAM photosynthesis. C4 plants, and essentially the goal of both of these, C4 and CAM, is to take CO2 in when plants can and store it in an organic acid form, so that it can then be used later when it has to close its stomates to increase the concentration of CO2. And when you increase the concentration of CO2, you can combat competitive inhibition. Keeping high concentration of the substrate you want compared to the substrate you don't want allows us to, at least, it won't be perfect because you're still building up oxygen, but it'll at least help to minimize the effects of photorespiration. In C4 plants, they're going to store CO2 in a separate cell. Okay? They're called mesophyll cells, which are on the top. They're going to store it as an organic acid called malate, and eventually they can take the carbon dioxide off of malate and put it into the Calvin cycle, which happens in a separate cell. So carbon fixation, when it first goes into oxaloacetate and eventually malate, is in a separate pathway. And notice it goes into a 4-carbon molecule. That's why it's called C4. A separate pathway than when carbon's actually finally turned into sugar in the Calvin cycle, which happens in a separate cell. So it separates it spatially. This is showing you, again, kind of where this happens in the bundle sheath cells of C4 plants. Calvin cycle in a separate place where they actually fix the CO2. In CAM plants, they're going to do the same thing. They're going to store it. Notice they store it as malate. But in this case, CAM plants, which stands for crassulacean acid metabolism, they store their CO2 only at night because they only open their stomates at night, because otherwise it's so hot in the desert. So they'll open at night, they'll store their CO2 at night, and then they'll release it during the day to do photosynthesis with. So they separate temporally. Instead of separating and doing this in different cells, they do it at different times of the day. Carbon fixation versus when they actually do the Calvin cycle. And this is a comparison of all three processes. Normal photosynthesis, where CO2 is directly fixed into the Calvin cycle. C4, where it happens in a separate cell, going on a 4-carbon compound first. And then CAM, where it happens only when it happens, carbon fixation happens only at night. And then during the day, they actually fix and put the CO2 through the Calvin cycle.